invite you all here for Talia's PhD defense seminar, the public seminar, and then we'll meet as a committee afterward, but we'll leave time for question and answers after her seminar. So Talia, I'm sure she sort of can't believe she's actually at this point. This is sort of the way it works, I think, after 6 years of work and trying to get your thesis written up and ready to be defended. It's always sort of a come down to the finish line and get it done. So she came to us from UC Berkeley with an interest in biomechanics. When I met her at a SICB meeting, it was sort of looking at locomotor stability in cockroaches, and she was working with another project on stabilization in falling geckos with Bob Full, and so I thought this would be a great opportunity to have a student interested in biomechanics. And then she came and she wanted to work on rhinoceros beetles. I said, well, I work on vertebrate animals. I don't really know about rhinoceros beetles. This guy, Doug Emlin at the University of Montana, you might want to consider. But really, we should think about some kind of a vertebrate organism, and ideally something that either flies in the air or moves on the ground. And at the time, we had a postdoctoral colleague, Kim Cooper, working with Cliff Taven, trying to understand the genetics of hind limb elongation, the evolution of skeletal elongation, and this interesting bipedal desert rodent, Jerboa, Jaculus jaculus. We had a breeding colony set up at the Concord Field Station, and I said, well, really, this is really a great animal to study, in terms of the broader diversification and what drives bipedalism in these desert rodents. And lo and behold, that caught her attention, and she ran with it. She ended up bringing in Jonathan Lossos as a sort of marrying the fields of biomechanics and morphology with evolutionary ecology. And I would say that one of the hallmarks of Talia's approach to science is that she is independent thinking, she's innovative, and she's very broad in the kinds of interests that she has. So she really has brought together a thesis that combines biomechanics and morphology, phylogenetic analysis, ecological niche modeling, where she analyzes climate, diet, or tries to analyzing climate, diet, and substrate properties in relation to also using information theory of use of entropy as a measure of unpredictability of movement in the natural environment, all in the context of predator-prey dynamics. So really a range of topics in the various chapters that are in her thesis. She's collaborated really broadly. That involves geographically going to Asia. She went to China with Kim, and then she worked here in North America, but she also got an Australian fellowship to work in Australia on the Tomies, partly with Chris Clemente, I think, and then some other colleagues in Australia. So she's done her PhD on 3 continents. She's broadly collaborated. She has a tremendous acknowledgement sections in her thesis. And I want to say that really, the golden egg that she laid for me was that she included Sadie, my dog, as one of my dog. The only time my dog has ever been acknowledged in anybody's thesis. Of course, she's not that old. But, maybe those of you that are finishing up with me might want to include. So along with the very cute and charismatic Jerboas, Sadie came to the thesis as well. So it really is, it's a treat to have Talia here to really, I think, pull together the range of things that she's been interested in. She's got a broad range of collaborations with an OEB, between the Lossos lab. There's a long list of people that influenced her thinking and approach to evolutionary ecology, along with those in my lab, and I think she's really interacted broadly with an OEB. I think benefited from the breadth of training that OEB offers. So with that, I'm going to let her talk about how you walk without rhythm. And for a guy who really doesn't dance that well, that's probably a good thing. And why, arrhythmic movements is a good thing from the point of view of a bipedal rodent. So, Talia.
So thanks, Andy, for that great introduction. Can you guys hear me? Is the mic on?
Yep.
All right, great. Thanks everyone for coming to see my talk. I know I may not be a familiar face for some of you. I've been away as a remote student for the past year, but I was here for 5 years before that, I promise. Let me go back to my clicker. So Walk Without Rhythm is actually a reference to the book Dune, in which the main character names himself after the hopping rodent of their alien desert planet. It's a lot like a Jerboa. So one of the most intuitive ways that we understand the way that evolution happens is through adaptation. And it's intuitive because we can all go out into the world and find these organisms that seem particularly well suited for their natural habitat. But how do we know that those organisms didn't just evolve those characteristics and they have nothing to do with survival in that habitat? Well, one of the hints that we can get that these traits and these characteristics are actually really useful for the survival in that habitat is through convergence. And that's when different animals that are really distantly related evolve similar characteristics in response to similar environmental cues. Now, this doesn't by any means guarantee that this convergent evolution is an adaptation, but it gives us a higher probability, it gives us good hints. And although adaptation is this really intuitive concept, it's actually a lot harder to pin down and really determine whether or not this trait is an adaptation or not. So I'm going to go through a very basic roadmap of how you determine whether a trait is an adaptation or not. And this is largely based on Stephen Arnold's 1983 paper, Morphology, Performance, and Fitness, but also if you want a much more nuanced and in-depth look at how to test these things, you should see Jonathan's paper in 2011 in the Evolution Journal. So the first thing I'm going to do is find out which traits are associated with this convergence, right? And then we're going to figure out what is the function of these traits. And then we can determine whether or not this function increases fitness. And this can be in survival or increase your likelihood of reproducing. And if we show this and then we show that it increases fitness in each of these animals in their natural habitat, it strengthens this convergence and kind of the cycle continues. So one of the most classic examples of convergent evolution is in the deserts around the world. You have bipedal hopping rodents. And bipedal means that they hop on 2 feet, usually their hind limbs, and they hop around in the desert. Now, there are 2 major ways in which this could have happened. Either there could have been one rodent that evolved bipedality and then it spread out to the deserts around the world, in which case it wouldn't be a convergence. Or they could have all evolved bipedality independently. So one way in which we can determine whether or not it's an independent convergence is by looking at a family tree. And let me go back a second. This family tree called a phylogeny shows us how related each of these rodents are to one another. And in red here, I've circled all the bipedal groups. And what we see here is that bipedality evolved independently 3 times. So this is a convergence, and it gives us a hint. It makes us a little bit more confident that it might be an adaptation. So let's take a closer look at these 3 groups. First, you have the Australian hopping mouse, and these animals are the most recent to evolve bipedality. And then in the center, we have kangaroo rats that live in California, and these live in California. And then in jerboas, they are the oldest group of bipedal desert rodents. And although I did work on all 3 groups of bipedal rodents, I'm only going to talk about jerboas today. So first, let's figure out which traits are associated with this convergence. How do we go from this quadrupedal rodent up here, and how does this have a wacky looking desert rodent that's bipedal down here? So the first thing that we wanted to do is look at the skeletal morphology and how they move around. So we took these high-speed 3D X-rays of jerboas running in the lab, and these are paramyscus mice that are quadrupedal on the right here. And what you can see is that in the jerboas, their legs stay directly below their body as they move around, whereas in these paramyscus mice, they kind of move more laterally side to side. And when we take a side view, we see that the jerboas, even though they're bipedal, they don't stand upright like a human being would. They still maintain their hunched posture. So this is really important for learning how these skeletons move, because a lot of times what you'll see in the museums around the world is that the jerboas are in these ridiculous postures because no one knows really how inside of this little puffball the skeleton is arranged. So together with Kim Cooper, Ferris Jenkins, and Laszlo Mazzoli, we we're able to create these postural reconstructions that are relevant to the way that they move. And we've got the Cecista betulina, the woodland hopping mouse, or woodland mouse up on the top, and then we've got our Jaculus jaculus, the jerboa down here. And so there are a lot of features that we can see that are different between these organisms. We've got different numbers of toes. We've got fusion of the central foot bones, and you can see, of course, that this hind limb has elongated greatly. So just seeing that there is a difference between them doesn't really tell us too much. What we want to do is look at the pattern of morphological evolution in this group, and that can tell us something about the process of evolution as well. So one of the first and main ways in which we can look at the pattern of morphological evolution in a clade is by looking at the fossil record. And all of the blue dots here show places where fossil jerboas have been found, but unfortunately a great number of them are tooth fossils, so they don't really tell us too much about hind limb evolution in jerboas. So instead, what we're looking at is the extant or the currently living members of the superfamily Dipodoideae. So there are 51 species in this group, 33 of which are bipedal jerboas. And here, on the left, we've got our quadrupedal and our facultatively bipedal jerboa, which means that it hops just every once in a while. And then over here in red, we've got all of our obligately bipedal jerboas. And this color scheme is going to be maintained throughout. So all of the more ancestral traits and all of the quadrupedal rodents are going to be in blue. All of the derived traits and all of the bipedal rodents are going to be in red. So what's interesting here is that there are actually 6 distinct morphotypes of bipedal jerboa. And this gives us a lot to work with when we look at the pattern of morphological evolution in this group. The first thing that I'm going to look at is digit loss. So that's loss of some of your toes. And this could be useful because losing your toes, which are really, really far away from your joint as you're swinging your leg, decreases your moment of inertia. So it decreases the cost that your muscles have to, the energetic cost of your muscles to redirect your limb as you're moving your leg. This is why it's easier to swing your arm when you're not holding a bowling ball. But the downside to this is it increases the impact of the stress per bone. So every time you hop, your body weight and the ground reaction force is divided by only 3 toes instead of 5. So you may be more prone to breakage. The next thing we're going to look at is metatarsal fusion. So your metatarsals are your foot bones, so kind of think these but in your feet. And in many jerboas, the central 3 foot bones are fused together. And this doesn't just mean that they're close together and they're kind of stuck together, but actually if you look here, in most of them, or in many of them, even the marrows are joined. So this is a partially fused Euchroides nassa, the long-eared jerboa, and it's got partial fusion, so it's 3 distinct bones at the tips, and then they've got an overlapping marrow in the center. Why is metatarsal fusion useful? Well, it actually has to do with the bending stress. So if you're jumping around on your toes and your body is supported up here, then every time you jump, you are giving bending stress, bending load to your metatarsals. And the way that we study and the way that we evaluate how well something is able to resist bending is by looking at the second moment of area. And that increases strength when you have more mass further away from the neutral bending axis. And so what we see here is that in each of these small toes in blue, and this is the cross-sectional view of the toes, when you have smaller toes, they have a smaller second moment of area, so they're more likely to bend and break in response to high stress, whereas this larger metatarsus that's fused actually has more mass away from the neutral bending axis, and so it is reinforced against this impact stress. So when we want to look at the pattern of morphological evolution of this superfamily Dipodoideae, the first thing we have to do is build a phylogeny or family tree. And I did this using nuclear DNA. So the way that I've arranged this tree is with the quadrupedal rodents on top, the bipedal rodents in red at the bottom. And also to note, in the bipedal rodents in red, in general they go from small to large. So first let's take a look at digit loss. Blue circles mean the ancestral trait, or 5 toes. Yellow here means 4 toes. And then red means 3 toes. And what we have in these smaller circles is the states of the extant, or the currently living species. And then what we've done is statistically reconstructed the probability of the morphological traits of the ancestors. So the probability that any of these ancestors are going to have a certain state is given by this pie graph. And what we see here in digit loss is that digits have been lost independently at least 3 times. And so this leads us to suspect that bipedality does not necessarily need to have digit loss, but it may benefit someone from digit loss. On the right side here, we've got metatarsal fusion. And here in blue, that represents unfused metatarsals. Yellow is partially fused, and then red is completely fused. And what we see here is a different pattern, where once fusion evolves, it's maintained throughout the rest of the jerboas. And remember, these are all much bigger than these jerboas up here. These are the pygmy jerboas that have unfused metatarsals. So this leads us to suspect that metatarsal fusion is probably a necessary state for bipedal locomotion as you get larger and larger and larger. So these discrete traits, digit loss and metatarsal fusion, are not the only differences between the quadrupedal and bipedal rodent. The most obvious thing is probably the length of these hind limbs with respect to these forelimbs. So usually we have a pretty good understanding of a lot of developmental mechanisms that make everything get bigger, but it's actually really interesting when we see some part of the body get bigger and the rest of the body doesn't get bigger. So we want to look at the ratio of the hind limb to the forelimb lengths. Right here, we've got the forelimb, which is all the arm bones, including the metacarpals, added up. And then we've got the hind limbs, which is all of the leg bones, including the foot bone, added up. And so the log of the forelimb and the log of the hind limb are here. This gives you an idea of how these scale together as you get bigger and bigger and bigger. So the blue line here is the quadrupedal rodents, and we've thrown in as well some mirrors, so like mice and rats and things. And you can see that all the quadrupedal rodents fit really well on one line, so they scale similarly. The hind limbs and the forelimbs maintain the same proportions as they get larger and larger and larger. But what we see here in red is that the jerboas have a different line. So they have a much longer hind limb with respect to their forelimb as they get larger and larger and larger. In yellow here, we have these transitional jerboas, the pygmy jerboas and the facultatively bipedal rodents. And remember this one spot right here. This is the Euphrates naso, the long-eared jerboa. And what I'm plotting here is, instead of pie graphs, because this is a continuous trait, we have a spectrum of color. And so again, we have blue is the shortest hind limbs, and red is the longest hind limbs with respect to your forelimbs. And what we see is that the largest jerboas are not necessarily those with the longest hind limbs with respect to their forelimbs. But in general, the hind limbs increase as you become more bipedal. Here we have the metatarsus to femur ratio, because we don't know whether the elongation of the hind limb is just due to just elongation of every bone, or if some bones are elongating more than others. And so right here we have the metatarsus on this axis and the femur on this axis. And again, we see that the quadrupedal rodents form this really great line, and that the jerboas, all of the derived jerboas, are on a separate line. The intermediate jerboas are in between, except for Euphrates naso, that one jerboa, the long-eared jerboa that I told you to look at before. That one is actually above the line. The other thing to note is that in both of these graphs, the intermediate jerboas are much smaller and the derived jerboas are much larger. So we suspect that in order to become bipedal, you must first become really small, and then as you get all of your derived traits, you can get bigger and bigger and bigger. On the tree, again, we have these quadrupedal rodents up here, the bipedal rodents down here, and again, in general, your metatarsals get much longer as you get more bipedal, but the largest jerboas don't necessarily have the longest metatarsals. This becomes really interesting when we look at both of these patterns together. We see that the patterns are not identical. So this is especially clear when you look at Eucherides naso, where it is one of the most derived metatarsal lengths, but it does not have one of the longest hindlimb to forelimb ratios. So this leads us to suspect that there are probably a complex suite of developmental mechanisms that are governing the evolution of these hind limbs. So there's not just one gene that says, all right, you're a jerboa. So, okay, we've got bipedal rodents with elongated hind limbs, especially metatarsals, metatarsal fusion, and digit loss. These are the traits that are associated with the convergence. Now we're going to look at whether or not the traits have a function. So this metatarsus to femur ratio is actually really informative when we look at the locomotion of animals as they across size, especially in mammals. On the left here, or actually on the very right, we have our graviportal animals that are trying to make their legs very columnar and really stand up and hold their weight up. In the center, you have cursorial animals that are specialized for running at high speeds over long periods of time, so they maximize locomotor economy. And then over here, you have your saltatorial animals and things with elongated legs. Okay? But what does saltatorial mean, and why is this kangaroo under both? Let's explore saltatorial locomotion with respect to cursorial locomotion a little bit more. So cursorial locomotion is, like I said, it is specialized for lowering your locomotor economy. And if anyone is playing full lab slide bingo at home, you can check that off. So all of these animals, no matter how many legs they have, no matter how big they are all dynamically similar. So their kinetic and their potential energy are in sync as they move along. Each leg or each set of legs acts like a pogo stick as they jump along. So this is where kangaroos fit in. They're saltatorial, but they're also cursorial. And this is really great because this means that their relative stride length to their dimensionless speed stays on this line, no matter what animal you are. And because we know that they're dynamically similar, we have this great equation that can help us predict when gait transitions happen. So when you have your Froude number, and the Froude number is about 0.5, you're going to have one gait transition. Your second Froude number of 2.5, that's going to be your second gait transition. And this works for all of the animals that they found that were cursorial. This even worked for the T-Rex when they were trying to figure out whether or not they would run at these speeds. So if we load in the hip height of a jerboa, we have a prediction of when they're going to transition between gaits. So the other great thing about jerboas with respect to the other bipedal rodents is that they actually have multiple gaits. So we suspect that they're going to hop until they reach about 0.56 meters per second, then they'll skip until they reach about 1.25 meters per second, and then they'll run. Sounds great. Let's test that. So this is what hopping looked like, and this is a gait diagram up here. So each one of these boxes is a stride, and the top line is going to be one leg, and then the other line is going to be another leg, and that's going to symbolize when they're in contact with the ground. So they hop simultaneously. All right. Skipping is kind of a new gait, and it's like a staggered gait where it's like hopping, but they have an overlap. And this happened in 80% of the trials. And then running, there's an aerial phase in between each jump, right? In between each footfall. This happened in 10% of the trials. And if we go back to our cursorial predictions again here, we thought that they would transition at these like really great lines, but what we found is that these means of each of their gaits are not actually statistically different. They are not significantly different from each other. And actually, hopping is used for higher, in general, a little bit higher speeds. Skipping is used for a huge range of speeds, and running is used at the lowest speeds. But then keep in mind again that these are not statistically different categories. But when we look at the mean acceleration, we do find statistical differences. We find that hopping is used for the lowest accelerations. Skipping is symmetrical about the mean, which means about 0, which means that it also it accelerates and decelerates. And running is symmetrical about 0, but there's barely anything there. So we think it's probably used to just maintain speed. The other thing we did when we ran these animals along a straight track is we put a force platform on the ground. And we got vertical and fore-aft forces. Fore-aft forces are going to tell us as well whether or not they're accelerating or decelerating. So here we have magnitude of fore-aft forces. Because these accelerations are so negative, I wanted to know whether or not hopping is used for both accelerating and decelerating. And when I found, when I did the magnitude of these values, I found significant differences. So hopping is used for changes in acceleration, and running is probably used for very little acceleration or deceleration. The other thing we found was really interesting. So it started off with a hop, and then it went into a skip. And that's what we call a gait transition. And this happened in 42% of all the trials. So all of the 80-10% that I showed you before, that was only in the single gait trials. This happened in almost half of all the trials. This happened so often that we were able to create a path diagram of gait transitions. And so we found that skipping was the one used most often, but it was also the one that was transitioned to and from most often. Hopping was preferentially used to stop, but we think it was also used to start, but our experimental system was biased. We had a window in the center where we were videotaping, and we'd scare them from either end. So we were able to capture many more stops than we were starts. So we have, I've showed you the kinematics of these jerboas, but now I want to go into the dynamics of these jerboas. So of saltatorial locomotion, so these hopping animals. There are two dynamical ways that you can be hopping. You can be cursorial, like the kangaroo, or you can be ricochettal, like the kangaroo rat. So we think that the jerboas are ricochettal. And our basis for understanding what ricochettal locomotion is like comes from Andy Buehner's work on kangaroo rats in 1988. So I'm going to talk you through the dynamical differences between kangaroos and kangaroo rats. So cursorial animals have low muscle contribution but high tendon energy storage and return. They're like rubber bands. They can store and return energy, and they have a low energetic cost for moving over a long period of time. Ricochettal animals have high muscle contribution, low tendon energy storage, and it's very energetically costly because muscle costs much energy. And the way this works is that imagine that this is your ankle, right? The white are your bones, that's your foot, white is your bone, and this is rotating about this ankle joint here. This is a muscle, and that's a tendon. So if you're cursorial, you probably have pretty stretchy tendons, and they can stretch back and forth like a rubber band. If it stretches a lot, you can move your foot, and then all of that stretching can be returned as energy in the next stride. Whereas for ricochettal animals, their tendons are not as stretchy, and so they have to move a little bit less, but then they don't get any tendon energy savings in return in the next stride. So how do we know how much the muscles versus the tendons are contributing to this locomotion? One way is to directly look with tendon buckles and get all of these direct measurements straight from the tendon and the muscle, but we did not want to do surgery on teeny tiny little jerboas that jump around and get tangled in things. So we did inverse dynamics. And the whole concept of inverse dynamics is that if you are standing in a certain posture, then the sum of all of the forces generated by your muscles and the moment that they create about your joint is going to be equal to the ground reaction force moment about that joint. And so we can set those equal and solve for the force that each muscle is generating, and then we can use that to determine the stress that the tendon is experiencing and how much it is lengthening, changing length. So because of all the variability that we saw in forward locomotion here, we decided to instead build a vertical track and have them jump straight up, because this is a repetitive behavior that they do in response to predators. So these jerboas were able to jump more than 10 times their hip height and often escaped. So we don't have any videos of that. And when we did these inverse dynamics calculations, we're able to see how much each joint is contributing to the overall jumping locomotion. So if we've got the hip right here, the knee joint here, the ankle, and then the metatarsal phalangeal joint, so the joint between your foot and your toes. When we did our calculations, we found that the metatarsal phalangeal joint is contributing the most, but also the ankle is contributing a large amount to this vertical jump. The other thing we did was look at the tendons. And so if we have a comparison of how much tendon energy savings different types of animals contribute to the total limb mechanical work, and if this is in cursorial animals that have forward locomotion, you can get a lot of tendon energy savings, right? And down here is the kangaroo rats that represent the ricochet shuttle animals that we have good characterized dynamics for. When we plot jerboas onto this, they're even lower than the kangaroo rats, so this is very low tendon energy savings in return. Not only that, we found a weak negative relationship between the amount that the tendon contributed to the total jump with height. And so the taller jumps, the higher jumps, use less tendon and more muscle, and the lower jumps use more tendon and less muscle. So now we have a really good way to characterize both the kinematics and the dynamics of ricochet shuttle saltation as opposed to cursorial saltation, which we understood from the kangaroos. So we've got short bouts of locomotion. These gaits have distinct dynamics, and they have frequent gait transitions. And everything that we know from the dynamics is also true. And so this makes us think that if you've got these gaits with distinct dynamic functions and you're constantly changing between these gaits, this is actually a specialization for maneuverability. So this is great. We've got the function of these traits. These bipedal rodent dynamics and kinematics are specialized for maneuverability. Next, let's look at how this function increases fitness. So it's all well and good to study animals in the laboratory, and this is really how we get these great, precise understanding of the musculoskeletal system, but these behaviors may not be relevant to what their survival in the wild, okay? So let's take a look at how jerboas act in the wild. This is a video of us catching a jerboa in its natural habitat in China, and you can see that it's running around. We're basically its predators at this point, frequent, moving around, and it's really difficult to catch, right? So this made us think that maybe this maneuverability is really useful for predator evasion, right? It also turns out that when we look at the ecology of these rodents, each of these bipedal rodents shares its ecosystem with a quadrupedal rodent. So they are burrowing animals, they come out at twilight, they forage for like seeds, roots, grains, and they all have the same predators, which are owls and snakes and things like that. So we can actually use them as a comparison, a relevant ecological comparison for looking at their predator evasion behavior, right? So if bipedal rodents are specialized for maneuverability, do these bipedal rodents actually have enhanced predator evasion in their natural habitat? So how does this work? What's the difference between maneuverability and predator evasion? Well, I think that the relevant metric for predator evasion is actually your predictability. So the great thing about these predators that these guys are hunted by is that they're ballistic interception predators. And this means that the owl is going to watch how the animal moves. So it's like, all right, you're moving this way at this speed, I got this. It's going to plot an intercept trajectory and then it's going to get its meal, right? But this type of ballistic interception predation is actually really susceptible to being tricked by unpredictable locomotion. So if an owl sees an animal moving, it's got this trajectory, and it plots an intercept course, but the animal moves unpredictably, then the animal's going to escape and the predator is going to go hungry. So we think that unpredictability in locomotion is something we can use to measure a predator evasion ability. And when I say unpredictability, I do not mean maneuverability. These are distinct things. So the way to think about this is that maneuverability is a metric of your intrinsic capabilities, and unpredictability is your behavioral utilization of those capabilities. And a good way to think of this is that maneuverability is what the manufacturer puts in your car. What is your car capable of doing? Whereas unpredictability is who is driving that car and what decisions does that driver make. I also don't mean variance. Variance is not a way to measure unpredictability. So this is a schematic of a little mouse moving around with a sinusoidal trajectory. It can increase the variance of its trajectory just by increasing the amplitude of its locomotion. Whereas an unpredictable trajectory looks a little bit more like this. And this figure comes from a 1985 qualitative paper just giving a rough drawing of a jerboa's trajectory. And if you're one of these rodents, I'll let you choose which trajectory you want to do. So we actually have, from information theory, a really great way to quantify unpredictability, and it's using entropy. This is the same metric used to measure information in a neuron. It's used to measure ecosystem diversity, and we can use it here to measure the unpredictability of these trajectories. I'm not going to go through the equation and how to calculate this with you right now, but I'm going to say that one way that we can visualize the data is through a distribution. And so the pointier your distribution is, the more frequent one thing is, the more predictable you're going to be. And so this means low H. Lower values of H are more predictable. Whereas the more flat your distribution is, the more likely you are to do multiple different things. And this means high H, and you're going to be unpredictable. So what I wanted to do was find out whether or not jerboas move more unpredictably in the wild than quadrupedal rodents that live in the same area. So I went to China with Kim, and we went to this area where there was one species of quadrupedal jerk and two species of bipedal jerboa. And this bipedal jerboa, Dicus sacchida, is actually really closely related and very morphologically similar to the jerboas we have in the lab, Jacculus jacculus. So we went to Xinjiang, China, in the desert there, and we took these videos. This is a quadrupedal jerk moving around, and we created these enclosures out in their natural habitat. And because these animals are more active at twilight and at night, we took videos at this time of day, and we scared them. So we simulated predator evasion and took videos of their locomotion. And this is a jerboa moving around, the same environment but from a different view. And because they used multiple cameras, we were able to track their movements in 3D. So these are the traces. These are not for the same amount of time, and the blue to red just shows you the directionality, and the space between the dots can tell you something about speed. So the further away the dots are, the faster they are. So just by looking at them, you can tell that these traces are pretty different. And when we do our entropy calculation, we find, yes, there are significant differences. But surprisingly, there are significant differences between all the species. So not only are the jerboas, the bipedal jerboas, significantly different from the quadrupedal jerk, but even the two bipedal jerboas are significantly different from each other in terms of the entropy or the unpredictability of their three-dimensional trajectories. But just having this number, this H number, doesn't really tell us too much about the types of behaviors and the type of locomotion that's creating these unpredictable trajectories. So what we did was we created these lower dimensional projections that together describe the locomotion of the animals. So up here in the top, we've got speed,
and then we've got probability. And so we see that this quadrupedal gird in blue here has the pointiest distribution, so that's the most predictable. So it's most likely to use these very low speeds, whereas the red jerboa here has the flattest distribution. So it's most likely to use many different speeds. Down here in the bottom, we have these spherical graphs that show the frequency of moving in a specific direction. And this is in 3D, so it's not just forward, backward, side to side, it's also jumping up and down. So what we see here is that the quadrupedal girds move forward and backward most often, and they rarely turn too much, and that they rarely move up and down. Whereas the Dipus sagitta here in green, they have a higher propensity to turn left and right, but also they move up and down a little bit more. But interestingly, the bipedal jerboa electica elater moves predominantly left and right and rarely goes in a straight direction, and it uses much more vertical movement. So this is how we can understand that even two bipedal jerboas can have very different unpredictabilities. So now that we've characterized predatory evasion and we've got this proxy in terms of entropy, does this actually increase fitness? Well, let's go back to this and think, okay, if these bipedal jerboas are the optimal solution for living in the desert environment, why haven't the quadrupedal rodents gone extinct? Why haven't they just been, outcompeted? Especially in these desert environments where the resources are relatively scarce. Well, I think that the bipedality and the enhanced predator evasion actually has something to do with it, because we know from various papers that predator evasion ability likely determines your foraging habits. So if you're really good at predator evasion, you're probably going to be more comfortable foraging in these exposed areas. Whereas if you're really bad at predator evasion, you're probably going to try and stay close to these covered areas where there are more bushes. And we know from trapping studies that yes, we're pretty confident that you're more likely to find quadrupedal animals inside of a bush, and you're more likely to find bipedal animals out in the exposed areas. But we don't know if this is just because the jerboas are trying to get towards the bushes, but the quadrupedal rodents are so mean that they're kicking them out, and it's this big struggle. Or the alternative is that the jerboas have actually evolved a preference for these exposed areas. So if bipedality enhances predator evasion by increasing the unpredictability of your escape trajectories, we would expect jerboas to be more, to feel less anxiety and to prefer being in these exposed habitats. And we can look to behavioral studies and behavioral techniques to determine this preference. So here we have the open field exploration test, where you've got a fenced-in area, and you've got a zone that is close to the area highlighted in yellow here. And if you've got a quadrupedal rodent that feels really anxious in these exposed areas, you're going to expect it to go straight for the side and kind of stick around close to the fences. Whereas if you've got an animal that is much more happy to be in the exposed areas, you're going to expect them to stay pretty much outside of that zone in the exposed area. So what I did was I took these same videos that I was scaring these animals in, and then I created this zone in each of the areas, and then I recorded the amount of time each animal was in or out of the zone. But this isn't a very controlled experiment, because we were constantly scaring these animals the whole time, and we were doing this over multiple days. And so, and also this zone, the width of this zone, it can be kind of arbitrary, and depending on the width of your zone, it can change your results. And so I did another test, which is called the light-dark box test, which is much less arbitrary. So you have a box where half of it is exposed to light and half of it is in the dark. And if you've got a quadrupedal rodent, you expect them to stay in the dark. And if you've got a bipedal rodent, you expect them to stay in the light. And so I performed this test. These are sped up, but this is the quadrupedal gird here, and then the bipedal jerboa here. That's actually moving. It's actually playing. And I did this in the lab. These are both lab-reared animals as well. And And we found really big differences between their movements, even under these controlled settings. So let's first look at the open field exploration of the videos that I took in the field. And you see that, yes, the quadrupedal jurors here are spending much more time in that zone close to the edge, whereas both gerbos are spending much more time in the center exposed. What's really interesting is if we plot this against entropy, we get a positive relationship, positive correlation, that the more unpredictable you are, the more time you spend out in the open. So that's great. That's very consistent with our hypothesis. Furthermore, in the laboratory environment, we see the same kind of pattern. The gerbos are going to be spending much more time out in the exposed and bright areas, whereas the jurors are going to be spending more time in these in the dark. So how could bipedalism increase fitness? What I've shown is that bipedality probably enhances predator evasion by enabling, and this enables a preference for exposed areas, and this reduces competition with other rodents. So they can actually spatially partition their resources and reduce this competition. And this may actually explain how you're able to have so many different species and so many different rodents, all depending on the same limited resources in these desert environments. So locomotion may actually be really important for community ecology. And this is great for when we want to think about how to manage and how to interact with these natural habitats. So this is my field site in Australia, and these are the people who run that site. And you can see that there are lots of bushy areas, lots of exposed areas, but by creating a road, they've created these huge exposed areas. And we know from the work that I showed that this makes it more suitable for bipedal rodents and not for quadrupedal rodents. Conversely, this is near my field site in China where they're trying to reclaim the desert and grow agriculture there. And while they're doing that, they're trying to green the desert, they're creating much more of these covered areas and leaving fewer exposed areas. So this is actually really not suitable for the jerboas and much more suitable for the quadrupedal jurors. So with that, I'd like to thank you for attending this seminar, and I'd like to thank these animals for being such an inspiration and being so quirky and diverse. I'd like to thank my committee, especially my two advisors. I found these really great photos of Andy and George from a while back, and I wanted to find other pictures to kind of match. I want to thank Andy and Jonathan really for allowing me to have this very independent and very weird thesis that's so different from both of their research. But they've been so supportive and really great to talk to for advice about all this. I really want to thank Stacy and George and Kim for being on my committee and having such good advice. Stacy and Kim aren't here right now. Stacy's at Davis and Kim just had a baby. So we thank them from over here. I'm going to unmute them for a second. All right. I also had a pretty rough time for some of my time in grad school. I lost a lot of important people. My grandfather is one of he's kind of this person in our family who really started us away from he started us more towards a logic and academic kind of idea, and my dad has fostered that in me. My friend Betty, my friend Jeremy, who we recently lost, are a big part of who I am, and I'd like to thank Farish for being such an inspiration, especially early on. They'll all be missed. I'd like to thank all the people at the Concord Field Station. They've been so good to me. It's been such a home away from home. And it really feels like home more than just a lab, and can get a little crazy sometimes. I'd also like to thank the Lassus Lab so much. They've really done a lot to turn my mind into this evolutionary thinking person rather than just a biomechanics, there is a form, there must be a function, must be an adaptation kind of idea. So thanks so much for being my friends inside and outside of the lab. I'd like to pay tribute to one of the greatest friendships of all time, me and Glenna. Without whom I would not have made it through grad school. And I want to thank especially all of my field assistants who have been so helpful and who have been just like amazing and overly qualified for everything in each of these countries. I couldn't have done all of this without them. I also want to thank the full lab. Kashik is here from the full lab. They created me into a biomechanist, and I signed up for blabbers. And I want to thank all these friends. I have a lot of people that I've been trying to stay in touch with over the years, both at Berkeley and around the world, especially Ruben from high school. And there's been just so many great people that have really helped me get through these past 6 years. And I want to thank my family. All of these people in Ohio have been sending me packages, sometimes anonymously, for the past month and a half. And it's been really great to get these surprises in the mail that are sometimes wacky, but always very, very amazing. And I'm so appreciative of them as a family. My main family, who I didn't know before I got here, and have turned out to be just such amazing people and have been such a refuge for me. My parents who are here today, and my family in Japan who are always there for me, even though they can't be here tonight. Ron is a co-author on one of my papers, and I was having a rough time being like, oh my gosh, these jerboas, I don't know what to do about them. Their locomotion is just so random. They're impossible to study. I wish there was just some way to characterize the randomness. And Rob was like, there is. So he really got me through the second half of my PhD. So with that, I'd like to thank all these wonderful people and then take any questions that you might have.
Okay, time for questions before we meet with her as a committee. I should have mentioned that Stacy from Davis and Kim from UCSD are actually with us. They're Skyping in. I think they're going to Skype in for the thesis defense. George is in the back there, along with Jonathan here. So, some questions before we.
Hi, Kitty.
So, given that this strategy of having an unpredictable movement is evolving multiple times, will you also expect that predators in those environments where those rodents are to evolve some sort of mechanism of outmaneuvering there?
Yeah, so the question is, I'm repeating it for the people in here. The question is, given that this unpredictable locomotion has evolved in prey so many times, do I think that the predators are going to evolve some kind of mechanism to counteract that unpredictability? So I actually think that that's one of the cool things about having these deserts where you have both quadrupedal and bipedal rodents. So if you have more quadrupedal rodents in these areas, then the predators are going to be used to the quadrupedal rodent locomotion, which is much more predictable. And this actually probably enhances the fitness of the jerboas, and their unpredictable locomotion being more rare, that they'll actually probably be more surprising to the predator. So that's why I think that they probably haven't had too much opportunity to evolve these different forms of predation. Thanks.
Yep. Go ahead. You handle the
Go
question.
ahead.
Let's see, it was a wonderful presentation.
Thank you.
And I could not help but notice in the last image of reclaiming the desert in China that was yet another allusion to Dune. But that wasn't my question. I guess I still don't have a question. I just have an observation for your second dissertation
Uh-huh.
that I'm sure will follow. It seems like a missing piece, or a piece that we'd like to add to the entire scenario of understanding why things are ricochettal and coexisting with quadruped, is to figure out what they're eating.
Yeah, so there have been, so the question was, can we figure out what they're eating, what these different bipedal and quadrupedal rodents are eating? I have a literature search on that, and it's in my thesis, so I didn't present it here, but it looks like the quadrupedal rodents may or may not be a little bit more broad in what they can eat in their diet diversity. So they are more aggressive when you catch them. So the way that I first started thinking about doing this comparative work across all of these deserts is everyone who's worked with these mammals in the deserts has come to me and said, oh, I love those hopping rodents. They don't bite you as hard like that. Those quadrupedal rodents bite so hard. And so this difference is maintained throughout all of them. So the quadrupedal rodents bite harder. They might have, consequently, a broader and more diverse diet. But I wasn't able to really establish that. I wasn't able to quantify the diet in a really great way in my thesis. But that's something I'm pursuing in the future.
If I can follow up, one is pressed to figure out if the ricochettal animal is staying in the open, where's the food it's eating?
Right.
And then another missing element that we probably don't think about often enough is how insectivorous are they?
Right, so the question was, where is the food in these exposed areas and how insectivorous are they? So just from my own personal experience, when I was performing these trials out in the desert, we had lights at the bottom, and they would sometimes attract insects. And so when you had a jerboa that would go through, they wouldn't even recognize these moths as food. But then the quadrupedal rodents, as soon as they found one, they just gobbled it right up. So I think there is a difference there. In terms of where is the food, it might be windswept, I'm not really sure, but one of the things I want to do for my postdoc is see what the animals are actually doing, where they're actually foraging. So I'm trying to set up this multi-sensor network to set in place in a community for a month at a time. Yeah. Hi, Judy.
I was just wondering, do the bipedals have a lower reproductive rate or a less number per litter than a quadruped, and maybe fitness and movement have something to do maybe with reproductive rate or number per litter or gestation length, something like that?
Yeah, so the question, the question was, do the bipedal and quadrupedal rodents differ in their reproduction at all? So I think Kim is actually much more experienced in terms of the reproduction of these animals than I am, but I haven't really actually looked too much at the jerks, the quadrupedal jerks. I think that in general, correct me if I'm wrong, Kim, but the litter size is generally around like 4-ish, but they range from like 3 to 6, I think, is the max. But what's interesting is that the Natomies, the hopping mice in Australia, there have been a lot of studies on sperm competition there because they have these boom and bust cycles, and they have to compete with all of these marsupials that have embryonic diapods. And so they may be different than the other rodents that are bipedal. But yeah, it would be a great study. Gotcha.
So you saw a difference in between the unpredictability between the different jerboa species. Do you have a hypothesis as to why that's the case?
So in terms of crown group jerboas, there are 2 crown groups. One of them are the 5-toed jerboas, and one of them are the 3-toed jerboas, and these guys have furry feet, and these guys don't have furry feet. And the 2 jerboas that I studied in the field, one was a 5-toed jerboa and one was a 3-toed jerboa. So I think it might have something to do with the groups that they're in. Oh, sorry, the question was, why are there differences between the 2 bipedal jerboas in terms of their unpredictability?
Any more questions back? Yeah.
Yeah, I just want
Or
to
is
follow
it, Jim, did you have a question too back there? Yeah. All right.
I was just going to follow up on that question. So is one fitter than the other? One seems to be better at ricochet locomotion than the other one.
So actually, I think that it's not that one is more fit than the other, but the fact that they have such distinct locomotor patterns. The question was, does that mean that one jerboa is more fit than the other? So I think that just because they have distinct locomotor patterns, that enhances everybody's fitness, because the predator is going to be looking at whatever's the most common, and if there are more things out there that are crazy and unpredictable, then the predator's going to tear its hair out and not know what to look for.
Talia,
Yeah.
why don't you also comment on microhabitat preference differences for the 3 and 5 toed animals? Because you talked about the fur on the toes, but they're not in the same locations either.
Right. So that's a good point. When we were in the field, we were able to find much more of the 5-toed jerboas on the flat, kind of hard-packed, crusty desert, whereas the 3-toed jerboas with the furry toes, they were much more on kind of the loose sand. So we did these trials in both microhabitats.
And one back from that.
I was wondering, you had the hind leg, right? The full leg.
Yes.
Oh, what about the full leg shrinkage? Is there any evidence of full leg shrinkage?
Yeah, so the question was, we looked at forelimb to hind limb ratio, but is that just hind limb growth, or is there forelimb shrinkage? And I think we saw some amount of forelimb shrinkage. So we did a lot of these morphological measurements, and then we did a principal components analysis. And aside from the first principal component, which we took to mean body size because it was in the same direction and same magnitude as all the measurements, the second principal component was metatarsus length and negative humerus length. So we think that there was some shrinkage.
Okay, well, let's thank Talia for a very nice presentation. One further announcement, I don't think there's any kind of formal anticipated celebratory party right after the defense. You're going to meet.
Is there?
Yeah,
Honda
there
is in
is.
charge.
Yeah, it's whatever time in the MCZ Labs tea room on the same.
Okay, well, that's where we're going to be. Okay, so I think we have to, one of us has to leave by about 5 o'clock, so we're going to have question and answer with Talia through about 5 o'clock or so, 5:15. So I knew there was going to be something done at the
At night, 7 p.m. at Parlor Sports.
Parlor Sports. I hadn't heard about something local to campus. And anybody that would like to crash, the field station is going to have a party for Talia tomorrow afternoon for lunch. So if you want to crash the luncheon party, we're going to have plenty of food. Feel free to do so.
Party all day.
Yeah. So, all right. Thanks for coming.
Thanks for